It is my pleasure to introduce to you today Dr. Shayna Kelly from UMD, who was also my supervisor, postdoc supervisor at University of Toronto. Dr. Shayna Kelly is a university professor at University of Toronto and a member of departments of chemistry, biochemistry, pharmaceutical sciences, and biomedical engineering. Dr. Kelly received her PhD from California Institute of Technology. She was an NIH postdoctoral fellow at Scripps Research Institute. And her research work is a variety of areas spanning biophysical, bioanalytical chemistry, chemical biology, and nanotechnology, and has pioneered new methods for tracking molecular and cellular analytes. Dr. Kelly's work has been recognized with a variety of these things, including Canada's top 40, under 40, NSERC, Stacy Flo, 2011, 2016 NSERC Brookhouse Prize. And she has also been recognized with ACS Inorganic Nanoscience Award, the Pittsburgh Conference Achievements Award, and Alfred Sloan Research Fellowship, a couple of Teaching Scholar Awards, and NSF Career Award, a new faculty award, and has been named Top 100 Innovator by MIT's Technology Review. Kelly is an inventor on over 50 patents issued worldwide. She's a founder of 3 molecular diagnostics companies, Genome Science, Exogenic, and Cellular Analysis. Kelly serves as a board director for Ontario Genomics and board trustee for the Flight Against Cancer Innovation Trust. She's also an associate editor for ACS Censor and editorial advisory board member for the Journal of the American Chemical Society and ACS Chemical Biology. And above all that, she's a great supervisor. And if you want to be successful in academia, besides interesting science that you will hear in minutes, you will learn different type of skills needed to be successful in this job. Let's say team management, personality, everything. So I'm very excited to have you here, Shayna, and thank you very much again for accepting our invitation. So please.
Thank you. I don't need, no, okay. All right, well, Sarah, I would say thank you for that introduction, but it's quite horrifying to have to listen to that. But anyway, I'm really delighted to be here today. It's always wonderful to be at McGill, but it is, you know, for somebody who spends their career training scientists and doing research, when you have people leave your lab and go off and do great things, it's really just one of the most wonderful parts of our job to see our trainees doing their own thing. And in particular, when somebody goes off into academia and then you see them starting their own group and training their students, it's just an amazingly rewarding feeling that you get. So I'm very proud of Sarah. I'm so delighted that she ended up at such a wonderful institution, and I'm really delighted to be here today. So I'm going to tell you about some of the things that we do today. As Sarah mentioned, we're a fairly interdisciplinary group working on lots of different types of projects. But one of the main interests that we have is developing new tools for molecular analysis. And we think about this type of problem from the perspective of developing new methods for the diagnosis of disease, also providing tools to clinicians that allow them to collect information that might be prognostic or predictive for their patients so that they can better use the therapeutics at their disposal to give their patients effective treatments. But increasingly, we're also looking at developing new tools for really discovery of new druggable pathways, discovery of new disease biology. And I was originally trained as kind of a hybrid physical analytical chemist, and I think in the analytical chemistry community, we very often kind of stop with we do the diagnostic angle of things, but we don't then kind of circle back to the biology part of how our tools can be deployed. And so that's a more recent direction that we're going that I'll tell you about today. And a lot of the problems that we try to solve are really just classic needle in a haystack type problems, where you may have a fairly complicated clinical sample or biological specimen. It may have billions of different types of molecules in it, or not different types, but billions of molecules that you need to sort through to find a very low level of, let's say, a target protein or a nucleic acid sequence that's going to tell you something about the physiology of a patient or the biology of a system that you're interested in studying. But so certainly these are very challenging analytical problems because the need and requirement for specificity is so incredibly high. So we've built a variety of systems over the years that are aimed at collecting this type of information. And I show a few of the main areas that we work in here. And many of the systems that we develop are based on different types of nanomaterials. So over the years, we've worked with semiconductor nanocrystals, quantum dots that we can engineer to have DNA molecules as ligands, but we also use DNA to help the quantum dots recognize things that we're interested in sensing. This is particularly powerful for either in situ detection, so if you're looking for something on a surface or like a hard surface or even the surface of a cell, these are good sensors for that application. We've also recently worked with these materials to make them able to go inside of cells and look at analytes intracellularly. We also have a project that I'll tell you a little bit more about today that is focused on a class of chip-based sensors, where we're really then using these sensors as a component of a device, where we're trying to analyze a sample for particular DNA molecules or protein molecules. And then I'll also tell you today about a fairly new area of work where we have a platform based on magnetic nanoparticles that allows us to profile cells and really be able to deconvolute the heterogeneity of cells that may look fairly similar phenotypically or genotypically, but have important differences in their actual molecular makeup. So these are some general areas that we work in, and then today I'll spend a little bit of time on our sensor work and then also on our magnetic profiling effort. But the system that I'll start with is a class of chip-based sensors, and working with these types of sensors and also really engineering them to have high levels of performance requires that you deal with a set of challenges that I'll try to lay out for you before I describe the work that we did. And basically, this relates to the fact that if you're going to make a sensor, you're going to have it, let's say, in some type of microfluidic device, sitting down at the bottom of a drop of solution where the molecules that you're looking to analyze are kind of floating around or diffusing around. You're limited by a few key things that have to be taken into consideration to develop high-performance sensors. And the important performance criteria here include sensitivity. So how low can we go in terms of the concentration of a target analyte? And we have to achieve high levels of sensitivity because we don't want false negatives, right? If we're looking for a particular molecule, we don't want to miss it. We don't want to say that the sample is negative when it's actually just a sample with a very low level of an analyte. We want a high level of specificity because we don't want false positives, right? We don't want to say that there's some pathogen present when it isn't actually, or it's some other, let's say, non-dangerous type of bacterial species. And then increasingly, speed is very important for these types of sensors. The trend is really to think about taking diagnostic instrumentation out of centralized labs or hospital labs, move it closer to patients. But as you do that, you need turnaround times for new diagnostic assays that are really minutes or maybe even tens of minutes, but they can't be hours or days, right? It makes no difference to have a type of technology that you can use closer to a patient, but it's still going to take days to get any type of result. So these are different types of performance that we need to keep an eye on. And so at a molecular level, and I'm a chemist by training, so this is really my perspective on these kinds of problems, that the things that we have to keep an eye on and then really engineer our systems around include being aware of molecular complexation kinetics and thermodynamics. In building these devices, we're going to be taking some type of a biomolecular receptor, a piece of DNA, an antibody, some molecule that will help us bind the molecule that we're looking for. And so what's going to limit the performance of our sensors is the kinetics of that receptor analyte complex being formed, and then also the thermodynamics, right? How stable that complex is. And so we have to keep a close eye on that. Also molecular diffusion. So in most types of samples, really all you can rely on to get a molecule to the surface of a sensor is just a diffusion of that molecule through a sample. Even if you use fluidic flow to try to speed things up, right, you're always going to be still limited at a surface in terms of how much transport you can actually have. And then surface area is another important parameter. Sorry, I should have been highlighting these as I go, because the surface area certainly drives your ability to have a molecule complex, or the probability of a molecule interacting with a sensor surface, but also in the type of electrochemical sensors we work with, the more surface area you have, the more background signal you're going to have, right? So it's a delicate balance. You want a lot of surface area so that your molecules can interact with the surface, but you don't want all the background noise that comes with having high surface area. So with all of these considerations in mind, the system that we've worked with over the last few years is focused on this class of nanostructured microelectrodes that are shown here. And so I have a cartoon version over here where you get the general idea. So these are our large three-dimensional structures that we fabricate using metal electrodeposition off of the surface of a chip. And then you can see the SEM image on the right. You can see these are really almost piles of needles, metal needles. And they achieve, and I'll take you through the data that supports this, but they achieve very high levels of performance for a few different reasons. One relates to having large surface area, but they achieve the surface area not through having a solid structure, but having these needles that then really distribute the surface area over a much larger amount of solution. And then as well, we can functionalize these sensors with, let's say, DNA molecules if we're detecting another DNA molecule in the sample. And the fact that we have this a high level of nanostructuring on the surface of these sensors allows the probes to be displayed in almost a native conformation. And that turns out to be very advantageous as well. And we couple these sensors with an electrochemical reporter system that I won't spend a lot of time on today because it's mainly chemistry more so than the types of topics that I think you'll be more interested in. But basically the way this works is that we have redox active ions that will bind to a complex that forms on the surface of our sensor. And that gives us a handle or a redox active signal that we can monitor to be able to determine whether or not a DNA sequence is bound to the surface of our sensors. Okay, so I'll give you a little bit more information about how we develop these and show you some of the data that backs up the points that I just made. And so basically, these are our sensors that we fabricate on the surface of a microfabricated chip. And the way that the chip is laid out is that we have these gold contacts that we pattern on the surface of, it can be any type of material, anything that's flat and fairly robust. So silicon works, high-grade glass works for this purpose. But we create this array of contacts using photolithography and putting down these structures made of gold. We then have these leads that are going to separate our sensors from the contacts. Because the contact, that's where we're going to basically put our alligator clip onto the chip in order to apply a potential and measure our current. And then we put a layer of silicon dioxide over the top of the chip. And basically what this does is it passivates the surface so that the leads are not talking to the solution. But instead what we do is at the tip of each lead, we put a hole in the silicon dioxide, and that creates an aperture for our sensors to grow when we use electrodeposition. And so as you can see on the right side, then what we're doing is plating metal into that aperture. So we're turning metal ions into basically solid metal by putting a potential on the lead that's at the bottom of the structure. And then what happens is that the metal will deposit into the aperture, it fills up the aperture, and then it spills over onto the surface of the chip. And when it does that, it gives us this fairly, or a nice high level of nanostructuring on the surface of a microelectrode. Okay, so that's how we make our structures. And what we did in the early days of working with the system is we explored a lot of the materials chemistry that you can use to create lots of different types of morphologies for these types of sensors, and also sizes. There's many different properties that one can fine-tune using electrodeposition. And I should mention that this was the work of a person who's a graduate student with me at the time, Leila Soleimani. She's now on the faculty of McMaster and running her own lab, and some of her activities are still focused on metal electrodeposition and making sensors, but she was the one who did this beautiful work about 10 years ago, where she showed that we could generate all these different types of sensors using one type of metal. So we used palladium, used the microfabricated chips that I just walked you through the layout of, and then she was able to generate sensors that were either very smooth, like this one on the left, which is basically a hemisphere of palladium. You're looking at it from the top. And then she could also generate sensors that were quite rough and had nanostructured roughness on their surfaces. And so she used these sensors and looked at their performance in a DNA detection assay just to get a sense of how the nanostructuring then affected detection sensitivity. And so she did this using an electrocatalytic reporter system that I mentioned a little while ago. You don't need to worry too much about the details, but basically, if a complex is formed on the surface of our sensor, meaning that one DNA strand has bound, so our target molecule has bound to the probe that's immobilized on the sensor, then we'll get a nice big electrocatalytic signal that we can monitor that allows us to track the overall efficiency of the sensing system. And what Leila found is that basically if we had very smooth sensors versus ones that had nanostructuring on the order of about 200 nanometers or had surfaces that were basically small particles that had a diameter of about 10 to 20 nanometers, that we could see the limit of detection for this system really moving around according to the surface morphology. And so shown in blue is the amount of electrocatalytic signal that we're seeing as a function of our analyte concentration. And you can see it's these sensors that can go really low in terms of the concentration of the analyte. Here we're in the attomolar concentration range and still able to get a detectable signal, whereas as the roughness of the surfaces decreases, you can see that the detection sensitivity also decreases. So here she was really able to do an apples-to-apples comparison of these different types of sensors, but again, same materials platform, same material, to show that there was a direct link between surface morphology and sensitivity. And with our best sensor, she was able to get down to a level of 100 molecules in a drop of solution. So a very, very low detection limit using this type of strategy. And so where we went from this initial work was to starting to think about different types of applications that we could target using these sensors. So we had the capacity to be able to detect specific sequences of DNA or RNA. And so you can look through the literature and come up with a wish list of different types of applications that you might be able to target using this type of approach. The most challenging application, at least that we could come up with, was to potentially look at a patient that had a blood infection. So bacteria actually in the blood. In this type of infection, you have a really low level of bacteria in the blood, so about 10 cells per milliliter of blood. You can look at other applications, for example, trying to determine whether someone's nasal passages are colonized with methicillin-resistant staph. This is a fairly significant problem. About 20% of us carry this antibiotic-resistant bug in our noses. If you're healthy and have a healthy immune system, it doesn't hurt you. But anyway, if you end up in the hospital or in ICU, it's important to determine whether you have this antibiotic-resistant bacteria present so that it can be controlled and you don't pass it to other patients, or it doesn't end up affecting you. If you were to swab somebody's nose to try to make this determination, that swab, once you eluted the material off of it, would have about a cell per microliter of bacteria. You can also look to see if somebody has an STD using a swab, but used in a different part of the body. There you would end up with about 10 bacterial cells per microliter. And then if you have a patient that has a urinary tract infection, their urine would have about 100 cells of bacteria per microliter, so a much higher loading. So you can take all of these different kind of bacterial loadings in terms of cells per microliter and then convert that into molarity by assuming that you are looking for a messenger RNA, one of the easier types of nucleic acids to look at in a real sample because it's already single-stranded, right? So you don't have to deal with getting a double-stranded molecule to dissociate. And so if you think about looking at messenger RNA and then you use a copy number of about 1,000, you'll end up with these concentration numbers that range from low attomolar to high femtomolar. So that's really the target concentration range if you want to look at real samples. And so we crunched these numbers. We took a look at the target concentrations. But what we also thought about was the fact that when we were transitioning from our initial experiments where we were just looking at oligonucleotides as we were doing our titration, a small piece of DNA and a buffered solution, now thinking about looking at messenger RNAs, what you realize is that you're dealing with a much, much larger molecule and a much more slowly moving molecule in solution. And so through some calculations that I won't take you through, we realized we needed to make our sensors a lot bigger. So we started out with ones that were just a few microns across. We realized that we actually needed to be about 100 microns across and 100 microns high in order to have a reasonable chance of being able to detect messenger RNAs on a reasonable time scale, so 10 to 20 minutes. So we went back to the lab and worked a little bit more on our materials chemistry and tried to make some large sensors using palladium. And what you find is that palladium electrodeposition, it doesn't really allow you to build structures that are tall. The palladium just crawls along the surface of the chip, and you don't get any verticality out of the structures. After a lot of searching around, we realized it was gold electrodeposition that would give us these long needles that allowed us to create really structures that had the 100 micron across footprint, but also 100 microns tall. And I wanted to mention one of the things that Sarah worked on with me when she was in my lab. This is kind of a funny story. So Sarah came to me one day and she said, Shana, I've been looking at SEMs of these sensors and I found something really interesting. I said, Sarah, what is it? She said, well, I looked at every single one of these needles and they all have 5 sides. Not 4, not 6. They all have 5 sides. And I kind of looked at her like, okay, you've been sitting at your desk counting sides of needles. I'm very happy for you. I'm very glad that you figured out they all have 5 sides and they're very consistent. They don't have 4 and they don't have 6, but that's great. But what are we going to do with this information? But Sarah was, she was way ahead of me. And she showed me this, and it was very convincing that you did have this really interesting, 5-sided geometry for all of the needles, but she had gone ahead and done a fib experiment that shows you why they have 5 sides. So you can see this 5-fold twinning inside of every needle, and that's what really then drives this pentagonal geometry. And through other work that Sarah did, what she realized is that these were really growth hotspots. So as we were doing our electrodeposition and really driving the kinetics of electrodeposition, the parts of the sensor that would grow the fastest were these 5-sided needles. And so they dominated the structure because of the electrodeposition conditions we were using and the kinetic control that we had over the deposition reaction. And so I'm sorry that I was skeptical, Sarah. It turned out to be a beautiful observation and a beautiful story that she was really able to flesh out by doing some really just very clever experiments. And she had kind of backtracked for us because we made these structures. We found a few years before Sarah was actually with us that these were really good for detecting bacteria, and we could go down to the levels that we were after in terms of those different clinical applications. But we had never really stopped to look at these structures and why they grew and what was interesting about them. And so Sarah was really able to highlight to us what was interesting from a materials chemistry perspective. And then over the years working with this platform, there's a few different reasons that I should mention that these sensors are able to achieve very high levels of performance. One of them relates to the fact that they're three-dimensional. And what's very advantageous about doing electrochemistry with three-dimensional sensors is that you can take advantage of radial diffusion, right? If you just have a flat sensor on the bottom of a chip, you're just waiting for linear diffusion to bring molecules to your sensors. Now, if you're three-dimensional, your molecules can benefit from radial diffusion, and our electrocatalytic reporter system benefited from radial diffusion. And the signals that we had to measure were then much, much larger because of that access to radial diffusion. So that's one of the things that makes these sensors special. And then also it is the large surface area, but it's surface area that's really dispersed through these long needles that we're able to create. And it turns out, I don't have time to go into this today, but all of the action in terms of the electrochemistry that's going on is actually happening at the tips of the needles. You have really high electric fields at the tips. That's really where most of the electrochemical current is coming from. And so rather than having a solid sensor that's giving us a lot of surface area, we almost have this distributed network of little points that are picking up our signal from solution. That's part of what makes these work really well. And then just a couple years ago, we did a set of molecular dynamics simulations that showed us something very interesting, and that we were really taking advantage of the fact that on our nanostructured surfaces, the probes that we were using to capture our target molecules, they were just in much more favorable conformations for forming complexes with the molecules that we were trying to bind. And so that effect also drives the performance of these, because when you tether a molecule to a sensor, very often its binding affinity just comes way down because you're tying one end of the molecule to a surface, and so it can't move around the way that it would in solution, and so it's harder for it to make a complex. But the other fact that is, or the other thing that happens that really makes it difficult for molecules to bind is that they're packed in a monolayer. They may not really even have the ability to see things that are in solution, but by putting probes on a nanostructured surface, you give them more kind of freedom to move around and sample the solution, but also interact with incoming molecules. So we've learned lots of interesting things through working with this sensor system. And then we've also pursued lots of different clinical applications using these as a platform. We focused in the early days on infectious disease just because of the time sensitivity of diagnosing infections, both for infection control, but also for better patient outcomes, just to get people treated effectively. And so we developed a variety of different assays for bacterial pathogens, also some viral pathogens. We spun a company out of the lab called Exogenic that built a nice desktop-based system that could be used to run this assay. The company was acquired by a U.S. defense contractor, General Atomics, that is now taking the units out into remote medical clinics for the military. And so the technology is kind of well on its way to making it towards the clinic. We've also collaborated with transplant surgeons interested in looking at molecular markers that can help predict the outcome of an organ transplant. And this is particularly important for lung transplants, where we only transplant about 15% of the lungs that are donated by people. And this it's a striking number because about 30% of the people who are on the waiting list to get a transplanted lung, they actually die while they're waiting. So we only use a very small amount of the supply, but then there are people that are losing their lives because there's just not enough organs to go around. Part of the reason that we only transplant about 15% is because transplant surgeons are very conservative. They have a lung that comes out of a donor, they're assessing it to look and figure out whether it should go into a recipient, and they're very careful because if you put a lung that is maybe pretty inflamed or just not in great shape into a recipient, the person may die, right? So you have to be very careful with those decisions. But the way that the assessment is done now is it's very general. There's different parameters that you assess, but the collaborators that we work with have found a panel of molecular markers that are associated with the outcome of how the lung does in the recipient. So rather than kind of doing the assessments based on the ability to hold oxygen, these kinds of things, we can look at the molecular level profile of a lung as it's being assessed for transplant using the technology that I just told you about. We also work with people that are interested in the bioproduction of therapeutic stem cells. We've built sensors that can be embedded into a culture system as these cells are being grown, just to help with the overall yield of these types of cells. This is quite important for regenerative medicine. And then over the years, we have collaborated with a number of oncologists, all of whom have their favorite cancer biomarker. They have a freezer full of samples that they really want to be able to crank through very quickly in order to know whether they have something that may be clinically relevant. And so we've done lots of these studies over the years where we've looked at different types of markers. But I'll use this as a segue to the other system that I wanted to tell you about, because what we found over the years is that one thing that our system wasn't, or this sensor platform wasn't very good at doing, was looking at circulating tumor cells. So these are cells that can be found in the blood of cancer patients. They're cells that break off from a tumor. And we had some collaborations around trying to analyze these cells, but we were getting very mixed results as we went along. And in looking more into both the biology of these cells and the clinical application of analyzing these cells, we realized that we had to get away from this type of sensor platform, which is really a bulk analysis platform, right? We have a sample, we have the molecules in that sample, and then we're doing this analysis. But if we have a mixture of cells that are actually maybe a little bit heterogeneous, not a homogeneous cell type, we're going to get mixed results from our platform because we're just doing this measurement where we're kind of blind to information coming from single cells. I'll tell you more about what I mean by that in a minute. But just to give you a little bit more background about circulating tumor cells, so they break off from a primary tumor. They're circulating around in the bloodstream, and then these are the cells really that are the most problematic for cancer patients, right? Most patients that are affected by cancer don't die because of the primary tumor, right? It's the metastatic tumors that arise that are really problematic. And so these cells are critically important for us to understand just in terms of their biology. How are they able to seed metastatic tumors? And we honestly don't know how that happens. It also turns out that these cells are very heterogeneous, and so you can take a collection of cells out of a blood sample from a cancer patient, and if you were to characterize them genotypically and phenotypically, you'd realize it's not just one type of cell that I'm looking at. There's cells with a variety of different properties, and there's a few different reasons that this heterogeneity arises. So for all of these reasons, we thought this was a problem that really needed a new type of platform or a new type of analytical solution. And the heterogeneity really comes from two different phenomena. So there's a static type of heterogeneity that arises because within a tumor, you can have different subclones. Right? And tumors are not necessarily homogeneous throughout. The vasculature of a tumor can be quite uneven, and so different parts of the tumor are going to have differing levels of exposure to oxygen, exposure to chemotherapeutics. And you may also have just different genetic clones within the tumor growing differentially, and so there's going to be different regions of the tumor that may have different phenotypic and even genotypic properties. But that's fairly static heterogeneity, right? So if a cell was to leave from a different part of the tumor, it may have different properties. But there's also more dynamic heterogeneity because as a cell leaves a tumor and is circulating around in the bloodstream, right, it's likely started out as part of an epithelial tissue, right? So it has epithelial properties, meaning that the cells are good at sticking to each other and they're good at being a part of a tissue. Once they're in the blood, they don't need the epithelialness anymore. So they start to change their phenotype, and they go into a state that we refer to as mesenchymal. And this is a state that is just, it's more characteristic of a cell on its own that's basically just trying to stay alive in the bloodstream. And the ability of a cell to change from being epithelial to mesenchymal, it means that it has a lot of plasticity, right? It can change its state because for metastasis to occur, then what has to happen is that this more mesenchymal cell has to find a place to kind of colonize that tumor, and then it's got to go back to being epithelial. Right? So these cells are kind of going back and forth between different states. And this is important because we think that it's these cells that are the most likely to have high levels of metastatic potential. They have to have this plasticity. So how do we track all this? I mean, it's bad enough, to have to solve a needle in a haystack problem, right? Where you have all these, all these pieces of hay and maybe just a few needles, and you have to, figure out how to sort through all the hay to find the needles. But what if the needle is changing on you? Right, as you're looking for it, maybe it's epithelial, maybe it's mesenchymal, it's changing its properties. Like how the heck do we build a tool that's going to allow us to take a look at all of this? So I'll just maybe take you through 10 minutes of our work on this problem. And our idea here was to build an approach that would allow us to deconvolute some of this complexity. And our initial idea was to go after proteins on the surface of the cell and on the surface of these circulating tumor cells, so that maybe we could start, just having the ability to profile the properties of these cells. And if you're familiar with methods like flow cytometry or FACS, right, this is the type of approach that we need to develop, right? In flow cytometry, you take a fluorescently labeled antibody, you bind that to the surface of your cells, you run the cells through a flow cytometer that then measures the amount of fluorescence, right? So it gives you a direct read on surface expression. But that's not a single cell level measurement, right? Flow cytometry is, as soon as you go below, let's say, 10,000 cells, you don't get much of a signal anymore. So we thought we need something like flow cytometry, but then it's going to allow us to analyze really rare cells. Because one thing I didn't mention is that in a milliliter of blood, you may have 2, 3, 4, 5, maybe 10 or 20 circulating tumor cells, if you're lucky, and over 1 billion blood cells. Okay, so you have a very small number of these cells to look at. So our approach, and so here we're trying to look at cells that may be the same cell type, but with different levels of a surface protein. Here I'm going to use as an example EpCAM, which is an epithelial protein that you find on the surface of epithelial cells. And so if we want to be able to distinguish cells that are, let's say, low EpCAM, medium EpCAM, high EpCAM, our idea was that rather than using fluorescently labeled antibodies, we would use magnetically labeled antibodies. And maybe we could label the cells at a level that would reflect the amount of protein expression on the surface of the cell. And we thought that was a good way to go, but there was a major problem that we had to solve. So the advantage of using nanoparticles for this type of application is that we could potentially load the cells stoichiometrically with 1 nanoparticle per protein molecule, if our nanoparticles were small enough, right? So we could then really get very precise measurements of protein expression. But the problem with nanoparticles, and if you're using magnetic nanoparticles as a label and you actually want to trap cells based on the number of magnetic nanoparticles, is that you really have almost no magnetic susceptibility to work with, right, to actually trap them. So we had to do some work on a microfluidic system that would allow us to work with particles that were so small. And what we ended up doing, this was the work of a postdoc, Reza Mohammadi, who developed these X-shaped structures that are good at, as a blood sample is flowing through our device, it slows the cells way down, but just transiently. So the cells are kind of interacting with that X as a barrier. They're slowing way down. They don't have a lot of drag force that's acting on them. And then even though we don't have a lot of magnetic susceptibility to work with, the fact that the drag force is so low means that if we have our magnets on top and below the device, that we can actually trap the cells, even though there's just a little bit of magnetism there that we have to work with. And so what we do is we process a blood sample to try to find those circulating tumor cells. We flush the device out. You can see there's a little collection of cells there. Those are the ones that we're trying to capture. And then we immunostain the cells just so that we can make absolutely sure that they're cancer cells and not regular blood cells. So we stain them with DAPI in blue just to make sure they have a nucleus. We stain them with cytokeratin to make sure they are epithelial cells. We also introduce a stain for CD45, which is a blood, it's a marker for blood cells. So we are just kind of making sure we don't have false positives there. And then we can count up the number of cells that we've trapped. Now we're going to read out how much protein expression using a device where we have our exterior magnets that we're going to use for trapping, but we also have interior magnets. So we make these out of nickel. And basically what we're doing is we're just making little circles, hopefully you can see these, around our X-shaped structures. And at the beginning of the device or the channel, the interior magnets are really small. So we're not going to get a lot of magnetic trapping force in the early zones of the device. But then as the channel keeps going, the magnets get bigger and bigger. So that cells that just have a few nanoparticles on their surface, or sorry, cells that have a lot of magnetic nanoparticles on their surfaces, they're going to stop early in the device. The ones that just have a few are going to keep going, but eventually they'll get trapped. Okay? And so then the way that we use this device is we take our blood sample, we flow it through, we process it, we visualize where the cells stopped in the device using immunofluorescence, and then we build a profile where we go along and we're imaging the cells using fluorescence and then just tabulating what zone of the device they ended up being captured in. Okay? And that allows us to build this type of histogram where we get a distribution. It shows us, how much spread there is in the level of protein expression. But we can also quantitate, are these high expressing cells, low expressing cells, how much heterogeneity, and then we can also get an overall count. How many of these cells were there in a milliliter of blood from a patient? And so we've showed that this works with culture cells, for example. In the top right, you're seeing the flow cytometry profiles of 3 different cell lines that have different levels of EpCAM. Here in our device, we're taking 100 cells, flowing them through, and then creating that distribution that I just walked you through. We can look at different markers. This method is not great when it comes to multiplexing because we can't really look at 10 markers at the same time. We can layer them on using immunofluorescence, but we're looking at one capture marker at a time. But we can take a sample, we can split it 3 ways and look at 3 markers, for example, HER2, EpCAM, EDCADHERIN. So we can build up lots of information this way. But the thing that's really key and very powerful about this is that we can take 40 cells, 20 cells, 10 cells, we can keep going down. There's a little bit of sampling uncertainty that starts to goof us up in these types of experiments. But you can see that profile is just spot on. It doesn't matter how many cells are there. We always create the same distribution. And so this allows us to do really high precision protein expression measurements, but with a really small collection of cells. And so we can apply this to look at changes in circulating tumor cells, let's say in an animal model of cancer. So here we have MCF7 cells. These are breast cancer cells. We generated 2 different versions of the xenograft. One where we treated the animals with estrogen, which makes the tumors grow much faster. Here we withheld the estrogen. And what you find then is that if we take the blood of these animals, serially monitor, and then look at the EpCAM profile, if you look at the upper right, you see that on the first time point, you may not be able to see this, the labels are a little small, but we can see on our first day of sampling, after 3 weeks after we implanted the tumor, we have a nice tight distribution of the CTCs in terms of their EpCAM profile. But then as we keep sampling, 10 days later, another 10 days, another 10 days, another 10 days, the distribution flattens out. So we start to see that heterogeneity in the cells. They're losing their EpCAM. And then you can see the cells are losing their EpCAM. So they're losing their epithelialness. So we can see that these are cells that are really dynamic. We see something different for the estrogen minus group in that we start out with that same tight distribution. Things flatten out a little bit. We see a little bit more heterogeneity, a little bit of loss of EpCAM, and then nothing. So these are our CTCs that are really static. And so then one of the most interesting things that came out of the study is that then after we were done, we analyzed these animals for metastases. These animals that had the very dynamic CTCs did have metastases. So those CTCs that seem to have a lot of plasticity seem to also be able to seed metastatic tumors. These animals did not have metastases. So those static CTCs just didn't seem to be able to do anything. So this lets us get at some interesting cancer biology, and we've also done a lot of work with this platform with patients. One of the things that we're working on now is looking at markers that you can find on the surface of CTCs that may tell you which therapy to use for a patient. In particular, we've been focusing on surface proteins that are relevant for looking at checkpoint inhibitors, which are very important for the treatment of lung cancer, other cancers. I won't go through this, but this is something that's ongoing. And then we've built now lots of different types of assays using the same type of platform, including assays that allow us to look at intracellular molecules. So the work that I just showed you, we were looking at a protein that was on the surface of a cell, EpCAM, but we've learned how to make probes that have nucleic acid molecules on them. We then have to permeabilize the cells in order to get the nanoparticles in. But we can do the same type of profiling where we're looking and separating our cells based on the number of nanoparticles that have bound. But here we're looking at an intracellular messenger RNA. And this is something that we've applied to looking at sequences that are important for choosing therapies for prostate cancer patients. There's a splice variant of the androgen receptor that if you have the splice variant, it turns out you won't be very responsive to certain types of therapy, so it's important to be able to detect. And then just very quickly, we've taken the same type of approach, and the devices that I've showed you so far are destructive, basically, right? We run our cells through, we process our sample, the cells are then stuck to the surface of the chip, right? We can pick them off, we can peel them off, we can do single cell, genomics, that kind of thing, but they're not that easy to handle. And so recently what we've done is come up with a version, so we call this technique magnetic ranking cytometry, and we've come up with a version of it that is basically non-destructive using something that we call prismatic deflection. And the idea here is that rather than having our X's and our nickel micromagnets, instead we have these guides that we make out of met glass, which is a nickel alloy, a cobalt alloy. And the cells, based on how many nanoparticles they have on their surfaces, they will flow along these deflection guides, but the guides keep turning so that the drag force gets higher and higher as the deflection guide proceeds. And so the cells then will actually fall off the guide at a point that tells us about their surface expression, and then they fall into these channels, and then we can just collect live cells. And this opens up a lot of different applications for us where you need intact living cells at the end of the analysis. So this just shows you the cells flowing along the guides. Here you can see the cells kind of dropping off because there's just too much drag force, and then they're dropping into these channels that we can collect the cells. And so we can do the same type of profiling that I showed you before, but here we can collect the samples afterwards. We get very high purity fractions from this type of approach. And then we're doing a lot of different things now in terms of collecting these different fractions, growing xenografts, doing all kinds of things just with the cells that we're able to isolate. And then I won't go through this because I know I'm out of time, but we're using the same type of approach to facilitate functional CRISPR-Cas9 screening, because if you think about doing a whole genome screen, you're taking a collection of cells, you're putting in that CRISPR-Cas9 library, and then you're generating a very heterogeneous collection of cells. And then we can start to sort them and do screens that are based on interesting phenotypic properties of the cells that are being engineered. We're also doing some screening of antibody libraries using a similar approach. But in the interest of time, I will just sum up. I mean, hopefully coming back to this slide that I showed you at the beginning, we have tools now from this work that are powerful diagnostics, just in terms of making a call on a type of disease or a subclassification of disease. We're working a lot on tools for prognostic and predictive information for clinicians for therapeutic decision making. And then this, stuff that I glossed over at the end is our attempt to maybe also put forward some tools for this discovery of new types of druggable pathways. So I want to thank everybody who's a member of the current group, and as well all of our alumni. I've been incredibly fortunate just to have so many talented people come through the lab. I mean, Sarah knows this. I just sit in my office and type on my computer, right? That's what I do day to day. Or I come places like this and tell you about what we do. But it's people like Sarah and all the people on this list that make the kind of studies that I've told you about today, make the results happen. I thank our collaborators and our funding sources. And again, thank you so much for having me today. And I really appreciate your attention. Thank you.
Very much, Shana, for a very interesting talk, like always. So if there's any question that we have, I think, half an hour for taking your question. Yeah, please.
Thank you for the talk. So in the first part, when you were using those metal star-like structures, you were actually pushing the boundary of the difficulty of labeling cells. So if I wanted to just do it with pipetting, I have to struggle pipetting because it's diffusion limited.
Yeah.
But I really like that idea. But in the second part, it's still super cool. But you still have that part that you have to label your cells out of your chip with the magnetic antibodies. Yep. So just what's wrong with it? If we look at the whole process together, isn't it? I know you said that if you put your antibodies on your structures, it's not as accurate, but isn't it more efficient altogether, considering the time that you consume at the beginning?
Yeah, it just depends how time sensitive you are, right? For the infectious disease stuff that I talked about at the beginning, time matters, right? What I talked about in the second part, tools for cancer diagnosis, management of cancer, all these things, time is less of an issue, right? Just because you want to take a sample from somebody, you take your time to analyze it, you're going to have them come back 2 weeks later, and then you're going to talk them through the results, right? So we pay a little bit of less attention to that, but I take your point that someday it would be nice to have all of these types of tools, very integrated, not people, getting carpal tunnel syndrome from too much pipetting and all that, but, one step at a time.
I don't need. Okay, yeah,
For the recording.
Shana, thank you. It really is a pleasure to hear you to talk about your research. So I have actually two questions, but I'm going to ask first one, and after leaving to the floor. And if we have time, I come back. First is about the first part of your talk about this nanostructure, and I'm most interested in you went very quickly actually to the application part, and for example, bioproduction of stem cells. I'm just wondering, it is a kind of customized, or how you make that? Or all of them, you put the different antibodies on this nanostructure. And it is kind of on demand. You produce your stuff, or?
Yeah, well, that's definitely the idea, to have it be on demand. The initial prototype that we made, so this was a collaborative project with Peter Zanstra, who's interested in the bioproduction of therapeutic stem cells, particularly blood cells, right? But the issue is that you really want to watch the culture progress, because the yields, supposedly, should be a lot higher, right? If you're able to kind of watch cytokines that are being produced as the cells are differentiating, and then you're feeding the culture appropriately, you're going to get to the end, right, with a much higher yield of the cells that are the target. And so the initial devices that we made, it's basically just an on-chip ELISA. It's not really anything that fancy, but we now have some sensors that I think are a much better fit for that application. They're basically reagentless. So they're not doing a traditional ELISA where you're capturing and then you're developing and all that kind of stuff, but it's just reagentless, so that the sensor is just watching the protein concentration, sending an input to a system then that's looking at how a culture should be fed. So it's still a work in progress. That was the initial stuff we did on that.
Thank you.
Thank you, Shana. That was an excellent talk. So my question is, so you're detecting sort of on the low levels of cancer cells. Were you able to, or did you try to fractionate some kind of tumors and try to see how robust the system in detecting a large number of cancer cells?
We've done some of that, and because we get very obsessed with the low end, but there's people who say, but I really want to take an entire tumor and I want to profile it in its entirety. So we've done some work on that, and the deflection device that I showed you is a much, much better choice for high cell numbers. And so depending a little bit on the profile of the target antigen that we're looking at, we can, in theory, process about 1 billion cells per hour. So we have high throughput. It depends a little bit on the details. So we've done some of that work with dissociated tumors, not as much as I would like, because I think it's really interesting to think about profiling an entire tumor, right? I mean, not that you would do that for a patient, but in terms of learning more about the biology of tumors. But it's something that we can do, and I think it's really it's an exciting area.
It could be a low-cost solution to doing these precision medicine genome, right? So yeah, as opposed to using hours and hours of flow cytometry.
Right. Exactly.
So, yeah. So, yeah. Okay.
Exactly.
Thank you.
There's no question from the floor. Okay, so my second question related is to the last part of your presentation about the intracellular trafficking of this nanostructure that you sent. But maybe I missed this part. But what is because you were talking about non-invasive cell sorting, and after you send these guys into cells, and how it's be translated to non-invasive, or maybe I missed just the link.
No, you're right. That the intracellular analyte assays that we have, they are invasive. Those cells are, I mean, we have to permeabilize them, right? So those are not going to be live cells that we're going to get back. We have to look at a surface antigen in order to have the live cells at the end. So that's a definite limitation. But that's a tough one, right? Yeah. If you have an idea for how we could do it, we'd be very interested, but it's a tough problem.
Shana, you had a spectacular lecture. I think everybody here shares it with me. Just a very, very small question. Exosomes, are you interested in them? Is your technology capable of solving some of their problems?
Yeah. I mean, exosomes, we do actually have a few papers on analyzing exosomes. The thing I get a little bit stuck on with exosomes, I mean, both of the technologies I've talked about do have applicability to exosomes, but it's just so much harder to know that you have an exosome, let's say that's from a tumor, or that's from this tissue, not that tissue, because you just don't have the ability to say, okay, it has this marker, it has this marker, it has this marker. It's in the blood. It must be a circulating tumor cell. And so the applications there are a little bit harder to pinpoint as a result. And so Sarah knows this. I mean, there are a lot of people in the group, oh, we got to work on exosomes. I'm like, I hear you, but I don't know. I just don't know quite how to crack that yet. But there's a lot of work that's being done on, how do you find the right markers, what is unequivocal in terms of when it comes to identifying exosomes. So I think the field will move, and then once the biology is there, then we can kind of catch up.
Question? So another interesting, actually, aspect about Shana's research, I'm engineer, material science and engineer by education. So when I learned about Shana's research on how basically using materials, nanomaterials, to do some real and high impact research in biology world, I was very inspired, and it's also inspired me during basically the research now that I'm doing confidently using and sticking to materials, nanostructures material. So it's very amazing to see how materials can come to life and bring us interesting application. So is there any suggestion that you have for future, maybe future direction that we can actually have in research in diagnostics and therapeutics?
Yeah, well, it's an interesting question. I mean, the advice I typically give people is you kind of have to decide whether you're an applied person or a more basically, basic science focused person. And there's huge benefit on being either side. But really, most of the people that I know, and I'm certainly this way, you're kind of inclined one direction or the other. And if you're somebody who really is interested in basic science, then just go and explore and discover new things and just, keep your eyes open. If you're more on the applied side, figure out what problem it is you're solving before you go into the lab. Right? Don't make tools and then figure out later what they're for. Right? I think like the exploratory stuff is great on the basic side, and then on the applied side, it just takes a lot of the front end, why are we doing this? What's the unmet need? What could we do if we did develop this capability? And so I think for people that are just getting going, developing new interests, that you kind of have to, I think, take a look in the mirror and say, my interests are here, and these are the things that I'm good at, and these are the kinds of problems that I want to tackle. Because I've worked on things over my career that were just a terrible fit for my skill set, the type of curiosity I have, and we just didn't get anywhere. I mean, it's really, the science really has to fit you as a scientist, I think.
Exactly. Any other question? Sure.
Hi, thank you. I have a question. In the first sensor you developed, you said that the electrochemical reaction occurs at the tip of the needle. Just wondering, how did you realize that?
Yeah, there's a lot of stories I could tell you about that. But basically, the way you can tell is that you can do very careful surface area calculations. And you do them over a time window where you can basically see the inside of the structure go dark, right? Because there's no diffusion to the inside. You can do that, yeah. Or even just very simple types of cyclic voltammetry experiments, those kinds of things. We've also done simulations of the electric field profile, and then you can just see it, and you can see the rates of charge diffusion being just so fast at the tips.
Using as well kind of big molecule to detect like DNA, RNA strands.
Yeah.
So do you have any problem with steric encumbrance in hindrance?
Yeah, and it's also, we've learned a lot, so we've applied these as well to looking at reactions that are important for renewable fuels, like CO2 reduction.
Yeah.
And there, I mean, you can just see from your turnover numbers and the diffusional profile that it's happening at the tips. So some of the work that has been more informative has actually been on that side of things rather than the bio side of things, because the simulations it's easier with the smaller molecules.
And do you have any problem with saturation after that of the probe? Because it's like covalent, it's not covalent, it's affinity bonding, so it doesn't just go like this. So you cannot make did you think about doing long-term measurement or more continuous?
We haven't done those measurements ourselves, but I certainly get a lot of questions about, when do you reach equilibrium with a system like this? Because eventually things will diffuse in. We've never had the patience or, I don't know, the right kind of folks around to do the experiments, so I don't know what the actual time scale is.
Okay.
But you should get there at some point.
Yeah. Okay, thank you.
Yeah.
The answers were very good, actually. It's a matter of time.
Right, yeah, we'll get there.
Thank you very much. Thank you again.
Thank you.